Hi everyone, I'm Naodini. Today I'm going to present our work on identification of radiomics associated with glioblastoma molecular subtypes. Outline of this presentation is as follows. First, we will share the background, the current issues, and the motivation behind this research. Then we will look at our dataset, the segmentation, and the feature extraction processes. Next, we are presenting the statistical analysis and the machine learning-based subtype prediction methods and the results. Finally, the discussion and the conclusion will be presented. Glioblastoma is a common malignant tumor with a poor prognosis. Despite the treatment followed by the diagnosis, glioblastoma patients have a median survival less than 1 year and a 5-year survival rate of 5%. This aggressive behavior of glioblastoma is mainly due to the molecular level heterogeneity and complexity. Thus, identifying the prognostic genetic biomarkers is essential to decide the appropriate therapies and treatments. In the past decades, many studies have focused on identifying these genetic markers of glioblastoma. Consequently, Verhaak and his team have identified 4 molecular subtypes of glioblastoma that have unique alterations in each subtype. These subtypes are mesenchymal, classical, proneural, and neural. However, recently the neural subtype existent has been revisited and removed as it has emerged through contamination with non-tumor cells. Although these subtypes were identified based on the gene expression profiles, gene expression profiling analysis cannot be routinely performed on glioblastoma patients due to the costs and the invasive approach. Further, due to the heterogeneity, these alterations might also vary within the tumor. Overcoming these limitations, magnetic resonance imaging is widely used in clinical practice as a non-invasive and low-cost diagnostic tool. With the advancement in the field of artificial intelligence, deep learning-based tools have been developed for segmenting the gliomas using MRI. This further has extended the area of radiomics through extracting features for diagnosing, predicting survival. These features can also handle the intratumor heterogeneity. Therefore, in several studies related to various cancer types, imaging features are utilized to determine the underlying molecular subtype without utilizing genomics. Now let us look at the dataset we used. For this work, we utilized the TCGA-GBM publicly available dataset with both MRI and genomic data. The T1 contrast and FLAIR images were downloaded and the gene expression profiles were obtained from a previously done study. The common IDs with both imaging and genomic data were filtered and 59 patient cases were left for the study. The MRI images were skull stripped and registered to match the metadata with the MICCAI Brain Tumor Segmentation Challenge, BRATS Challenge dataset information. For segmentation, the BRATS 2018 dataset was used for training the deep learning model. For segmentation, we utilized 3D unit architecture with the 3D attention module that we proposed for the BRATS Challenge 2019. The input was a 3D MRI volume sized 155 by 155 by 240 pixels. As we mentioned before, this model was trained with the BRATS manually annotated data. Then the TCGA dataset was given to obtain the segmentation feature maps of the 59 patient cases. As shown in the image, 3 regions were segmented. The red region is known as necrosis, the yellow region is the enhancement, and the green region is the edema. Segmentation was followed by the feature extraction. The intensity and texture-based features, kurtosis, the histogram of the oriented gradients, entropy, mean intensity were extracted for the necrosis, enhancement, tumor core, and whole tumor regions. These histogram features were obtained from both T1 contrast and FLAIR images since both of them magnify different subregions of the tumor. For an example, T1 contrast provides a clear picture of the enhancement region. The extracted geometric features included the major axis length, minor axis length, centroid coordinates, inertia tensor, eigenvalues of the inertia tensor. Moreover, the volume features were extracted with the bounding box method as a fractal dimension. These features were obtained from the necrosis, tumor core, and the whole tumor regions. Totally, 119 imaging features were extracted from the MRI images. Next, we will look at the statistical analysis tests used in this study. Our initial experiments are to find the associations between genomics and radiomics. Thus, we utilized the Pearson's correlation to find the association among imaging features and the gene expression levels of glioblastoma patients. Further, we used both feature types, radiomics and genomics, separately to find the correlation with the overall survival of glioblastoma patients. Nevertheless, univariate Cox proportional hazard analysis was performed to identify the radiogenomic features associated with survival. Next, we focused on identifying the imaging biomarkers associated with the molecular subtypes of glioblastoma. First, to clarify the normality of the radiomic features, the Kolmogorov-Smirnov test is utilized, and the features with p-value less than 0.05 are identified as features with a normal distribution. Accordingly, we performed the Kruskal-Wallis non-parametric test to determine the significantly different imaging biomarkers between the four subtypes with the features that are not normally distributed. Additionally, to identify the subtype pairs that are significantly different, Wilcoxon test is applied between each pair. The Bonferroni correction is performed on the multiple comparison tests to avoid the family-wise error rate. Now let us look at the results of the statistical analysis tests. Out of 119 radiomic features extracted, 2 radiomics showed a significantly low positive correlation with the overall survival in days. These 2 features are the curtices of the enhancement region extracted from T1 contrast and FLAIR MRI modalities. These 2 features also gave statistically significant negative coefficients for the Cox proportional hazard as an analysis. Thus, the large curtices of the enhancement region is associated with the longer survival. The correlation between the gene expression level of 1,740 genes and the overall survival had also been analyzed. 25 genes were significantly correlated with the overall survival. These features were recognized with a hazard ratio over 1 and p-value less than 0.05 with the univariate Cox proportional hazard analysis. Besides, the correlation between the imaging and the genomic markers were also assessed. Thus, the expression level of TIMP4 gene showed a moderate positive correlation with the fractal dimension of the necrosis region. Similarly, the necrosis region's extent had a moderate positive correlation with the ACOX2 gene expression level. In order to identify imaging biomarkers associated with molecular subtypes, initially the Kolmogorov-Smirnov test was applied to obtain the normality of the radiomic features. 38 features out of 119 radiomic features were significantly normally distributed. Thus, for the remaining non-normal features, Kruskal-Wallis test was applied. Therefore, 12 features that were significantly different between 4 subtypes were identified. These features were include the fractal dimension of the necrosis, whole tumor, and the tumor core regions. This suggests that these features were significantly different between some subtypes. To identify those differences between subtypes, the Wilcoxon test was utilized pairwise. The fractal dimension of the necrosis, tumor core, and the whole tumor region showed a significant difference between proneural and classical subtypes, and also a considerable difference between proneural and mesenchymal subtypes. Moreover, the fractal dimension of the whole tumor showed a substantial difference between classical and mesenchymal subtypes. Now let's move on to the subtype prediction with machine learning methods. We used the significantly different features between subtypes to predict the subtype using machine learning algorithms. All the features were standard normalized before training the learning algorithm. Linear kernel function of the support vector machine, radial basis kernel function of the support vector machine, polynomial kernel function of the support vector machine, random forest classification, decision tree, and logistic regression were utilized for predicting classical, mesenchymal, and proneural subtypes separately as a binary classification with imaging biomarkers. First, the mesenchymal subtype was predicted with the aforementioned machine learning methods, and the polynomial SVM outperformed the other algorithms with an accuracy of 82.4. Next, the classical subtype was predicted and the linear support vector machine outperformed the other three algorithms with a four-fold validation accuracy of 85.3. Nevertheless, the prediction of proneural subtype was performed to obtain a four-fold accuracy of 83.1% with radial support vector machine. The subtype prediction with genomics was executed as a generalized method for predicting any of the four subtypes. However, since the neural subtype no longer exists, this method might require slight changes in the current exploration. Thus, the linear SVM gave the highest accuracy of 94.91 with the four-fold cross-validation. Our results showed that the kurtosis of the enhancement region has a positive correlation with the survival. The enhancement region had shown relationships with the survival in previously done glioblastoma-related studies, proving our observation. Another major finding of our study is the significant difference in fractal dimension between four subtypes. Despite the technological advancement, obtaining a consistent gene expression profiles is challenging due to the technical shortcomings. Therefore, identifying the subtype with radiomics helps the clinicians to determine the subtype precisely with the acquisition of MRI that has been used in clinical practice commonly, followed by fully automated segmentation and feature extraction. In this study, we address a solution for subtype prediction without employing high-dimensional genomics, which is costly and inconsistent with platform acquired. We identified distinctive radiomics between subtypes that reflects the underlying molecular level gene alteration between the subtypes. Accordingly, we developed a subtype prediction model with machine learning for glioblastoma patients. In conclusion, our work delivers a promising subtype predictive model that will lead to a better clinical maintenance of glioblastoma patients. These are our references. Thank you very much for your attention.